friends, welcome to the Biological Life Sciences channel. Today in this video we are going to see in details about the topic that is lactose operon. Okay, so Lac Operon or lactose operon is also called as Lac Operon. Okay, so in short it is written as Lac Operon. Okay, so we are going to see in details about this here in this video. So first of all, let's see what is operon. So operon is always a cluster of different genes. Okay, so there may be cluster of 2 to 6 genes together, in a single promoter. So what happens here in operon? There will be 2 to 6 genes, or more genes can be also there. So all these genes will be under the control of a single promoter. Okay, so whatever the mRNA it is going to form, the mRNA will be a polycistronic RNA because there is number of cistrons linked together in a single gene. Okay, so let's see in details about the Lac Operon. So first of all, we know that the lactose is a disaccharide. So it's a disaccharide made up of two different components. So let's first see the structure of lactose before studying the lactose operon. So let me show you the structure of lactose first, and then we are going to discuss in detail. So first of all, lactose, it is a disaccharide. Okay, it's a disaccharide made up of two different entities. So let's see what is the two different entities in lactose. So it is galactose and glucose. It is made up of galactose and glucose. Okay, so first of all, let me draw the chemical structure of the lactose as well as lactose, that is glucose and galactose, that is linked together in this. So the structure is like this. I will draw all the hydrogen bonds in this structure with a different pen so that you can be able to differentiate here. So there is a presence of beta-1,4 galactosidic bond here, glycosidic bond, and there are number of hydrogens and oxygen, hydrogen and hydroxyl group present here, as shown in the figure that I am drawing here. So this is the structure of lactose. Okay, so this is the structure of lactose. You can see there it is disaccharide. The first structure is here is of galactose, and the second structure is of glucose, and when they attaches together, it forms lactose. Okay, so lactose is a disaccharide sugar. Okay, so now remember, whenever the lactose is present in the medium, what happens? First of all, the cell tries to take the primary source of energy. Okay, so if the glucose is present in the medium and lactose is also present in the medium, the cell will prefer glucose over lactose. Okay, so first of all, the glucose will be used up, and then when the glucose finishes, then when no other energy sources is remaining, then this lactose is hydrolyzed by using the Lac Operon that is present in bacteria. Okay, so this operon system is generally present in bacteria where 2 to 3 genes or even more genes also are linked together and are under the control of a single promoter element. Okay, so lactose operon is one such example which was discovered the first time. Okay, so first operon that was discovered in bacteria, actually this operon system is present in bacteria, and this is very important in bacteria because it helps in conserving the energy, and then bacterial system is a less complicated system, so this kind of system helps bacteria to be less complicated and use less amount of energy. Okay, so let's see what is lactose operon. So lactose operon or Lac Operon was first time discovered by two scientists. One is François Jacob and Jacques monad. Okay, so these two scientists discovered the Lac Operon first time in 1960. Okay, so for this discovery they have been awarded Nobel Prize also. Okay, so for this discovery they had been awarded the Nobel Prize also. They deduced the mechanism of Lac Operon, and first time they showed that there is a presence of operon system in bacteria in which different kinds of genes are clustered together or linked together. Okay, so let's see first of all the operon system in details. So what are the different constituents of operon system? So remember, operon has always two elements. Okay, so operon system it has two elements. The first element in the operon system is regulatory element, and the second here is the structural genes. Okay, so regulatory element means the elements which helps in regulation of the operon are called regulatory elements, and the elements which is helping in forming different enzymes, actual enzymes are present in the structural genes. Okay, so regulatory genes can be repressor or operator region or any kind of enhancer in such kind of operon, enhancer, or it can be a attenuator region also. Okay, as present in tryptophan operon. Okay, so all these kinds of elements that helps in regulation of Lac Operon or in any other operons is called as regulatory element, and then the structural genes are there which codes for the enzymes that helps in the pathway for synthesis or utilization of a pathway of synthesis or utilization of a particular substrate of which the operon is. Okay, so in case of Lac Operon, the structural genes are going to produce the genes which helps in utilization of lactose. Okay, so let's see the different genes that are present in Lac Operon and what is its function and what are the products that the genes is going to form, that we are going to see now. Okay, so let's start. Okay, so let's see with first the structural genes that are present in Lac Operon. There are regulatory genes also. There are two regulatory genes here, that is Lac I gene that codes for repressor. Okay, then operator O gene, O region is there that codes for operator, and there are three structural genes that are called as, first is Z that codes for the enzyme, that is a specific enzyme, that is beta-galactosidase. Beta-galactosidase, it is the gene is named as Z. The next gene is Y, that is galactoside permease. And last is the A gene that codes for thiogalactoside. Thiogalactoside transacetylase. Okay, so these are the three structural genes that are present in Lac Operon. First is beta-galactosidase that helps in breaking down of beta-galactoside, breaking down of lactose. Okay, then second is galactoside permease. Okay, so remember this enzyme is a permease enzyme that is generally present in membranes. So this enzyme helps to transport the lactose from outside the cell to the inside the cell. Okay, so this is the function of the Y enzyme, Y gene here. And the A gene, last one A, it codes for the enzyme thiogalactoside transacetylase the breaking of the lactose molecule cannot take place without the addition of acetyl group. So therefore this helps in acetylation action of that particular lactose molecule. Okay, so let's first see the structure of the lactose operon, and then I'll be explaining you all the details regarding this operon. So generally there are three operator regions here. That is, three different regions are present in the Lac Operon for operators. So let's see the regions one by one. Okay, so suppose this is the double-stranded DNA, which I have shown in unwinded form just to explain you the different operator, different regions here. The first region that is present obviously on this side, on the right side will be the ZYA. Okay, so this is the Z, this is Y, and this is A. Okay, these are the structural genes, and then there is a presence of operator 3 region here in the beta-galactosidase gene, and there is a main operator, that is operator 1, present in the promoter of beta-galactosidase gene. So this is the gene for the promoter that starts before the beta-galactosidase gene, and there is a presence of Lac I gene here on this side. Okay, so in Lac I gene there is another promoter called as O3. There are three operator, that is O3. So there are three operator regions. This element that is present prior to the gene, structural genes, is called regulatory element. And the elements which are present after the regulatory elements is called structural genes. Okay, so what happens here, remember, this is the place for the binding of operator, this is the operator region. Okay, so now what happens, that I'll show you, suppose this is the +1 site, suppose from here the gene, actual structural gene is starting, means what, the gene is starting from +1 site, that is ZYA. So from +1, I'll tell you the region or the sequence where this operator 3, operator 1 and operator 2, this is operator 2 that is present. Okay, so let me explain you the first regions. So O1 operator is present right at the start site of ZYA, that is the +1 site. So it is present at the +1 region. Then O2 is present at +410 region, that is present in beta-galactosidase gene. Okay, so the O2 operator that is present in beta-galactosidase gene, the position is 410. So from +1 site up to from +1 site till 410 there is beta-galactosidase gene, and there is the presence of operator 2 here in between. And the next is operator 3, it is present at -90 site, which is prior to the +1 site, ahead of the operator 1, and it is present just after the lactose I gene, that is Lac repressor gene. Okay, so this is the positioning of the different genes in Lac Operon here. So first of all, what happens here, the Lac I gene, it codes for a repressor. Okay, so it's a tetrameric repressor, remember. Suppose this is a tetrameric repressor. Okay, so now what happens here, this tetrameric repressor binds to the operator 1 region. Okay, remember, it binds to this region. Okay, so when lactose is not present in the medium. So if the lactose is not present in the medium, this Lac I, and here I forgot to mention, here there is a promoter for lactose, this repressor. Okay, promoter for Lac repressor, and this is the promoter for Lac Operon. Okay, so the promoter starts from here and ends till here. Okay, so now, and this is the promoter for the repressor gene. So what happens, the promoter is transcribed and the repressor element is formed here. You can see the repressor protein, it's a tetrameric protein. Okay, so remember, it has four identical units here. what it does, it goes and binds at the operator 1 region. So what will happen here when it binds to the operator 1 region? It will entangle the DNA. Okay, so the operator 1 region is having maximum affinity towards this gene, that is, sorry, this protein, that is Lac repressor. Okay, so this protein is called Lac repressor. Okay, so this Lac repressor is going to bind at the O1 region and it is going to block the promoter for the ZYA gene, that is the structural genes for lactose operon. Okay, so what it is going to do, it is going to block, plus it is having the maximum affinity towards O1 region, that is operator 1, and it is having less affinity to O3 and O2 region. Okay, so what is going to do, what this repressor is going to do, it is going to entangle this whole operon like this. Okay, so you can see here at this position the O1 region is there and it is going to bind O3 and O2 also to this same protein that is present here. I'll show you like this. Okay, so this is the supposed tetrameric region here. So it is going to entangle this whole double stranded DNA together at this position, so that the O3 comes over here, O2 comes over here, and this protein is right at the middle. Okay, so thereby blocking the lactose operon here in regular scenario when the lactose is not present. Okay, so now when the lactose is present in the medium, okay, remember lactose directly is not utilized by this medium, so lactose for this Lac Operon is acting as an inducer. Okay, so means what, when the lactose is present, then only the ZYA is going to form. Okay, but by default there are fewer molecules of ZYA also present in the medium, because when this repressor protein binds at this position, it does not block the ZYA gene synthesis totally. Okay, only few molecules of ZYA, that is beta-galactosidase, permease and trans-acetylase are forming here in the constitutive stage. Okay, so very few molecules will be there. So now when the lactose is available, what will happen, the available amount of permease will transport the lactose in the medium. So lactose here is acting as an inducer. Okay, so lactose will induce this molecule and it is also the substrate for the same operon. Okay, so what is going to happen, lactose is acting as an inducer here and this lactose will go and bind to this promoter. Okay, so sorry, this Lac repressor, suppose this is the lactose and it has binded to this repressor and it changes the conformation. It changes the conformation of this Lac repressor in a such a way that it is not able to bind at the operator region. Okay, so due to the failed ability of the lactose repressor, when it is bounded to the lactose, this shown in green color here, is not, there is a conformational change and it is not going to bind at the O1 region, which makes the O1 region vacant for RNA polymerase enzyme to come and bind at this promoter region and synthesizes the ZYA gene. Okay, so when the ZYA gene is formed, synthesized or transcribed, mRNA is going to form here. The mRNA will be a polycistronic mRNA. This is 5' to 3'. So this mRNA is going to have all the three genes. The first gene is beta-galactosidase enzyme. Then second is permease. And the third is trans-acetylase. Okay, so all these three enzymes are going to be formed here in this way. So now when the three enzymes are formed, then the function of utilization of lactose is carried on. Okay, so one more thing is there, and then this Lac repressor is not going to bound here. Okay, again when the lactose is vacant, amount of lactose in the medium finishes, then what is going to happen? This lactose is the more amount of repressor is formed and no more lactose is remain to add to the extra repressor that is formed in this position. So the repressor goes and binds to this position and shuts off the operon. Okay, so this is how the regulation of lactose operon takes place. Remember, there is one more thing here, that is the presence of secondary metabolite, that is cyclic AMP, that is CAMP. Remember, cyclic AMP, it is adenine monophosphate in the cyclic form. This is also called as hunger signal. Okay, so what happens when there is a less amount of energy in the cell and there is lot energy is required, so the cell produces cyclic AMP, that is the cyclic form of adenosine monophosphate. This is acting as a hunger signal. Okay, so what this does, the cyclic AMP makes a protein called as cap binding protein. Cyclic AMP binding protein, cap binding protein. What it does, this cap protein, due to the cyclic AMP signal, comes and binds at this position. I'll show you. Comes and binds right at this promoter position here of the lactose operon. Okay, so this is a cap protein along with cyclic AMP. Okay, so what it does, when this cyclic AMP along with the cap protein binds at this position along with the RNA polymerase enzyme, this helps in increasing the fold of concentrations of this different enzymes. Okay, so there is about 10 raised to 5-fold increase in the synthesis of beta-galactosidase, then permease and transcristalize enzyme when all this moiety, that is RNA polymerase, cap and cyclic AMP, binds at this promoter region. Okay, so this is acting as another enhancer for this operon, and then when cyclic AMP and cap is not present, then the synthesis is about 10 raised to 3 folds. Okay, so if there is the repressor binding at this position, there is synthesis of only about 2 to 10 molecules only of all the 3 enzymes. Okay, so this is a basal rate at which the transcription takes place. So here the requirement is not there, but at least few of the genes should be present inside the cell. So therefore the transcription proceeds at a very basal rate when the repressor is binding at this position. Okay, so once the repressor is removed, the cell starts to synthesize all the 3 enzymes, and if the requirement is acute for the energy from this lactose, so at that time this cyclic AMP and cap protein binds at this position and ZYA genes are synthesized in a very fast manner. The synthesis can reach up to 10 raised to 5 folds, and then from the glucose and galactose that is formed, the glucose is taken for the utilization of energy, and then galactose, which is formed here in this operon, is again converted to the glucose, and then this glucose is utilized for the production of energy. Okay, so this was all about the regulation of Lac Operon. What I told you at the starting here, the lactose is acting as an inducer as well as substrate here. Okay, so there are 2 different molecules, that is IPTG and XGAL, that is analog of lactose. Okay, so that looks like lactose. Okay, so that is used to induce this operon, and the other one is to take or to be used as a substrate. Okay, so I am going to discuss that structures in details. First of all, I am going to tell you how the lactose directly is not utilized here. Actually, it's the allolactose. So first of all, lactose is converted into allolactose by the beta-galactosidase enzyme, and this allolactose is acting as an inducer here, not actually lactose. Okay, so what is the difference in the chemical structure that we are going to see here of the two? So first I will show you the structure of allolactose. What is the difference between the lactose structure and allolactose structure? Lactose structure I have already shown you in this video earlier. So now I am going to draw the structure of allolactose now, so that you can understand how the lactose is converted into allolactose, and this allolactose is used as a energy source. Okay, so let me first draw the structure. So this is the structure here. Actually there is a change in bonding here, you can see. So okay, so this is how it is going to form a bond here. So this is the structure of allolactose. This is allolactose structure. Okay, so the bond, that is beta 1,4 linkage, is modified by the beta-galactosidase enzyme, and this lactose molecule, that is a disaccharide, is converted into a allolactose, which looks like this. So now this allolactose is acting as an inducer, and this binds to the repressor and changes its conformation. Okay, so this is a regular inducer of the Lac Operon. We are going to see now the artificial inducer and the substrate of Lac Operon, which is used in variety of genetic engineering experiments, where you can utilize the Lac Operon or lactose for different purpose, for detection or screening of recombinants and all. So one of the famous experiment is the transformation, where you detect the recombinant cells or transformed cells using blue white screening. So in that case, there is a chemical called as IPTG, that is isopropyl thiogalactoside. This chemical is an analog of beta-galactoside sugar, that is galactoside sugar. So how it is used here, I will draw the structure of that first. There is a presence of sulfur here in this. Okay, so this is the structure of isopropyl thiogalactoside. So it is acting as a isopropyl thiogalactoside. Okay, so this is acting as a inducer. Okay, so this is the inducer for lactose operon, but the thing is, this cannot be used as a substrate. Okay, so when you want to induce the operon, or when you want all the three enzymes from the operon, then you can use IPTC. This is an artificial inducer that is going to induce the operon, but it is not going to act as a substrate. Okay, so this is used in variety of applications. Now we are going to talk about another chemical that is acting as a substrate for lactose operon, but that is not the inducer. Okay, so these are the artificial compounds that is an analog of lactose. The next compound is X-gal. I will write its chemical name. It is 5-bromo 4-chloro 3-indol beta-galactoside. Okay, so this is the chemical name of X-gal, that is 5-bromo 4-chloro 3-indol beta-galactoside, and its structure looks like this. There is a pentameric ring, and to this pentameric ring there is an attachment of a hexameric ring to this, like this. Okay, so there is a presence of NH at this position, and there is a presence of Br here and Cl group here. Okay, so now this regular bonding of HOH and here CH2OH NH. Okay, so this is the structure of X-gal. So now what happens when beta-galactosidase acts on this? Okay, so I already told you that this is a substrate here. It will not act as an inducer. So this does not have the ability to act as an inducer, but it can act only as a substrate. Means what? The enzyme beta-galactosidase can break this substrate. Okay, so when this breaks, or when the enzyme breaks this substrate, then a blue colored product is formed. Okay, then a blue colored product is formed. So this blue colored product helps in indication of a particular presence of particular gene or not in that cell. So here, therefore, X-gal can also be used in different cloning experiments, where you want a color differentiation between the different colonies of bacteria. Okay, so the most important application of this X-gal and IPTG is, we can see in alpha complementation, that is blue white screening in case of transformation. So likewise, this Lac Operon or the different enzymes in Lac Operon or genes in Lac Operon can be utilized for the detection of detection in lots of cloning experiments or genetic engineering experiments. Okay, so this was all about the details of the lactose operon that we have seen in today's video. If there is any question, please write your questions in comment box. If you like the video, please press like button, share, comment and subscribe the channel. Thank you for watching. Thank you so much.